we move on with the next speaker. He's also studying psychiatric conditions in non-human primate models, and Dr. Robert Desimone. He's director of the McGovern Institute at MIT, and he used to be a director of the intramural program at NIMH, so a real expert in psychiatric disorders, and he will introduce yet another model on autism, the Shank 3 model. Thank you.
Okay.
That's the red one, and that's the green one, which is a bit lighter.
Okay.
And the green one is if you felt like
Okay, let me.
green one. You can use the small blue one.
Okay, but it's not showing. My computer says it's, oh no, duplicated. Okay, all right. Okay, well, thank you, Nikos. Thank you, organizers, for having us here. This has been a wonderful event, a great celebration of the work that Nikos has done, and I think, as I think we all indicated last night, the best is yet to come. And we all expect to be invited back for another symposium 5 years from now, celebrating some of the great new success of this fantastic group that you have here. So I'm going to talk about some of our translational work in monkeys, trying to create monkey models for psychiatric disease. But it's actually a relatively discrete study. But after this discussion yesterday about whether how much motor behaviors and so on and state influence the kinds of data we get from monkeys under very controlled conditions, I thought it would be useful if I spent just a few minutes talking about the work we were doing on microcircuits in monkeys and the important role that that's playing in the attentional modulation of visual responses in monkeys. So I'm going to get a little detour, and then we'll come back and I'll talk about the transgenic monkeys. So the basic idea is that when those of us we study visual search, we're used to seeing eye movements roughly in the sort of theta frequency range, constantly around scenes. Monkeys are always directing their gaze at interesting things. But we and many others have been working for years under the assumption that we could eliminate these roles of eye movements by having the standard fixating monkey condition, where we'd have them fixate a spot and attend to things elsewhere, and so eliminate the eye movements as a factor and just manipulate attention. So that was the question, is actually, have we actually done that, succeeded? And so we began looking more closely at data from a task that's described here, which is a task in which monkeys started the trial by fixating a spot or monitoring fixation with the video monitoring technique. And then there was a little spatial cue, a very dim bar, pointing at what will be a future stimulus that appears on the screen. An array of stimuli appear, and the monkey is trained to attend to the stimulus that had the pointer, and then after some random period of time, it changes color, and the monkey makes an eye movement to the stimulus. And if you do a conventional analysis of firing rates of cells under those conditions, you see what everyone sees, which is that in the attended condition, there's a higher firing rate with a single stimulus in the receptive field, a higher firing rate when the animal's attending to it than when it's ignoring it. And you see that here from the in the histograms with the red being the attended condition, the blue the unattended. This is at the beginning of the trial, and this is at the end of the trial at the color change. So that was all well and good. But we do know that even when fixating, people and monkeys are often making tiny little eye movements around the fixation spot. And in the past, when we've looked at these, I would say we've seen them. We could find intervals without them, and the results seemed to be the same. And so we didn't worry about them very much. But we had a postdoc come to our lab who did his PhD work in microcircuits. He was an expert in both detecting and analyzing microcircuits. He was looking at our microcircuits very carefully. And first thing he noticed is something that, in retrospect, shouldn't have been surprising, which is that when you have stimuli that normally appear in an array with a certain configuration, the microcircuits are oriented towards and away from the attended stimuli in the array. So if the attended stimulus is here, the microcircuits tend to get oriented towards and back for that attended stimulus, likewise for the horizontal, likewise for the vertical. So they weren't random eye movements, they were directed eye movements. And these are also made, I think interestingly, in a sort of theta range, around 3 or 4 of these microcircuits a second. So same sampling frequency that you see under free gaze. And then the other thing that, although we had looked at the role of microcircuits previously, we did something in this analysis that we had never done before, which was to separate out the data according to whether the microcircuit was being directed towards the cued stimulus or back from the cued stimulus. And when we did that, we discovered something phenomenal to us, which was that when you separated out the data for microcircuits towards the cued stimulus, which could be either inside the receptive field or out, the response to when the microcircuit is being made to the attended stimulus was greatly enhanced compared to when the microcircuit was being made to the ignored, the uncued stimulus. Whereas when the microcircuits are being made away from the cued stimulus, there's essentially no firing rate difference. So in other words, all of the attentional modulation that we had seen in our conventional analysis of taking the whole trials into account were just averaging over these periods. And there was actually a very specific interval when the modulation occurred, and it was a much bigger effect when you restricted your analysis to just that interval. Now, an obvious question was that when you make microcircuits, there's actually a change in the visual stimulus. And so we ran a control where we mimicked the movements of the stimuli that are made by the microcircuits, but without the microcircuits to the stimuli because the animal wasn't doing the task. The animal's just fixating. And when you mimic that microcircuit-induced movements of the whole stimulus array, you didn't see virtually any change. So it was something related, presumably, to the correlated discharge of the command for the monkey to make the eye movement that was playing a critical role. Now, when people do these experiments, as I mentioned yesterday, there's a lot of variability when you see responses of cells. And this has been dealt with extensively recently in the monkey visual world. And one of the things that we have always noticed is that when you cue monkeys in tasks, there's a lot of variability in when they make their responses. If you look across all trials, you see this the heightened response when animals are tending stimulus versus ignored. But we decided we would we would look at these responses depending on when the microcircuit occurred during the trial. So we sorted trials out according to whether the microcircuit was made early, after the stimulus onset, or the cue, later, or later. And this is variability under the monkey's control. But then the question was, what was the effect of that on the neural response? And it turned out that this variation, this modulation that you see with attention to the stimulus, simply tracks the latency of the microcircuit. So what we thought had been just normal neuronal latency, variations in latency and noise and so on, was actually explained by the underlying motor behavior of the animals. It's also, we looked at the animals' latency to detect the stimulus change on the trial, and we did that for intervals if the change occurred when the microcircuit was being made towards the attended stimulus versus back, and this is just the cumulative latency. The red line is the with the circuit towards the attended stimulus, and the blue where it's made back. And so behaviorally, the animal's also faster at detecting the change if he's about to make the microcircuit towards that stimulus. Now, so these were based on recordings in V4, but we also had simultaneous recordings in inferior temporal cortex. And we saw something very similar there. If we looked at firing rates, it's more complicated there because the receptive fields include all the stimuli. But if you simply looked at firing rates towards the attended stimulus versus away, you see the same enhancement only on with the microcircuits towards the attended stimulus. We also had simultaneous recordings in the pulvinar during this time. And in the pulvinar, again, we saw the same thing that we're seeing in the cortex, which is higher firing rates only on with this attentional modulation, but only on microcircuits directed towards the acute stimulus, and essentially nothing on the microcircuit in return. If we did an analysis of decoding of the stimulus properties based on the cell's responses in both V4 and IT cortex, and that's shown here, we found that we could decode the identity of the cued stimulus much better with the microcircuit directed towards the stimulus than on the microcircuit directed away, and likewise, much better in IT cortex. And some of this decoding improvement is coming just from the enhanced response. But we're now going back and looking at these data even more closely, and we believe that there are additional enhancements to the neural representations of the stimuli on the microcircuits directed towards them compared to away, and those are contributing to the decoding performance. Okay, so that was the detour, and now we're going to come back to the what I was going to talk about, which is the development of a primate model in the autism spectrum disorders. And you heard a lot about that from Mu Ming. As many people know, autism spectrum disorders are of high prevalence. They seem to be even getting higher. They occur early in life as a neurodevelopmental disorder, and there's no effective treatments. The studies that have looked at the genetic vulnerabilities in autism over years, you find more and more genes identified which can contribute to the vulnerability to develop autism. The vast majority of those genes have small effects, so those are polygenic forms of the disorder. But there are a few genes that are highly penetrant and lead to a high prevalence of autism based on mutations. And one of those is the Shank 3 gene mutation, which exists in various forms in human patients, but the most severe forms are the ones where there's a loss of function. And those are the ones that we've tried to reproduce in the monkey using a knockout. In humans, the humans with a Shank 3 mutation have a syndrome known as Phelan-McDermott syndrome. It's typically a heterozygous mutation, leads to a global developmental delay, absent or delayed speech, hypotonia, lots of autistic-related behaviors, decreased perception of pain, mouthing, chewing non-food items, stereotypies, and so on and so on. And so the question was, could we both develop these primate models and then test them to see if they had behaviors that had any relationship to what are found in human and patients. Now, the Shank3 gene is widely expressed in postsynaptic synapses of excitatory cells, and so you would expect that the gene might have effects on lots of systems, and we believe that's the case, even in the peripheral nervous system, as recently shown by David Ginty. And this has been, it has been modeled in mice. So there have been actually quite a few studies looking at mice with Shank3 mutations, and they do have behaviors that could be argued have some relationship to those in autism. In mice, you normally only find those behaviors in homozygous mutations, which don't exist in people, and it's very hard to relate those behaviors to what you actually see in people. But for us, most importantly, that if you restore the Shank3 expression in these mice, some of these behaviors recover, which gives us optimism that if we were to use a monkey model to develop a gene therapy for the Shank3 mutation, that it might also help in some of the behavioral symptoms. So as I said, there's been a lot of work in mice in Shank3, but as you all know, there's big differences both in the evolution of the mouse versus the primate and the development of the rodent versus the primate brain, and particularly in areas that we think would be important for the highest levels of human cognition in prefrontal cortex, for example, medial prefrontal cortex, involved in social behaviors and so on. The prefrontal cortex is sort of rudimentary in mice. It does exist, but it is greatly enhanced and elaborated in all the primate species, including macaques and marmosets. So we now have published our first paper on these animals with the Shank3 mutations. This is a collaboration between some of us at MIT, particularly Guoping Feng, with our collaborators in China. China's labs are the world leaders in the development of transgenic primates, and I'll talk at the end about more of who our collaborators are, but we have a particular collaboration with the Shenzhen Institute of Advanced Technology, and this work could not have taken place without that collaboration. So we used CRISPR-Cas9 technique to knock out the Shank3 gene in these animals, and it was verified with protein expression. We had, I think it shows it in the next slide, we had 5 mutants. 2 of them were essentially homozygous, 3 were essentially heterozygous, but we were reporting the results from all of them. And so one of the first things that you notice when you look at these animals is they develop stereotypies. In mice, there's usually just one stereotypy that develops, this obsessive grooming. In monkeys, we see a whole variety. It's not just one stereotypy. Just a couple examples here of one monkey seems to have a compulsive finger licking, and another monkey has cage flipping. Other monkeys seem to have bar licking. They have their own idiosyncratic expression of these unusual behaviors, which occur, as you see, more often in the mutant monkeys. They have reduced vocalizations, and reduced social behaviors when tested in a variety of ways, including, I think, Mooming showed you an example of testing animals with probe animals, and we've done the same. And it's very apparent then in testing them with the probe that they have reduced social interaction. These videos just will give you a sense of that. They're going to show two monkeys in two separate cages with a divider between them. These are two wild-type monkeys, and the divider's being pulled out, and you'll see two monkeys meeting each other for the first time. Checking each other out. Okay, so then we'll go to the. We now have, you can see the mutant monkey with the cage flipping. Now the divider is going to be taken out. Now, you see, these are all hand scored by blind observers, and there's a clear difference in the lack of in the reduction in social interaction that you see. This is all now moving into automated classification of behaviors, which are also finding big differences in how the monkeys interact. So there's reduction in social behaviors. Another very common problem reported in autism spectrum disorders are sleep problems getting to sleep, problems going to sleep, again, as we heard in Moo Ming's presentation. And if we have activity monitoring of the wild type and mutant animals, it shows that basically in the wild type animals there's a pretty strong differentiation between night and day and the amount of activity, whereas in the mutant animals there's less differentiation. Looks like they're sleeping more during the day and sleeping less at night. And if you have quantitative measures of these disturbances, you can see these differences: slower latencies, more fragmentations of sleep, less efficient sleep. So it seems to mimic that aspect of the autism syndrome. Now, we did have them tested by having them look at either faces of other monkeys or other objects, and we did not see a difference. They did look at faces. In fact, in some cases, they looked more at faces than other objects. But what we did find was that while they looked more at faces of other monkeys, what they showed a really pronounced lip smacking when they did it, which was almost never seen in the controls. But these monkeys were lip smacking all the time. And lip smacking in the monkey world can mean a variety of things, but one of them is a kind of social anxiety submission. And we think, our anthropomorphic interpretation is that they were fixating because the other monkeys made them really nervous. But in any case, that was the behavior we saw. We tested them cognitively. They, on very simple tasks like a black-white discrimination, reversal, they were normal. But I tested it on a Hamilton search test like Moo Ming described in his monkeys. I'm not going to go through all the details of this, but successive levels of reversals in the spatial memory task. Basically, only one animal made it all the way through the test, whereas all of the controls did. And so this is early days, and we know we need to do much more cognitive testing in these animals to understand what their problem is, but it's at least encouraging that there's some sort of cognitive problem there. And we did MRI imaging on the animals, and If we look at the strength of connectivity, both the global and local connectivity, the global connectivity shows a reduction in the mutants for the cingulate cortex, motor cortex, anterior cingulate cortex, and this is found in some studies of autistic patients, this general reduction in global connectivity. Whereas if you look at local connectivity, then we just see, it's not one pattern, but you just see differences from normals. So in the thalamus, there's less local connectivity, less in the striatum, but much more so in somatosensory cortex and in visual cortex. And it is at least possible, and this is just hand-waving, but it is possible that this could be related to the hypersensitivity that is sometimes described in people with Phelan-McDermid syndrome. So as Muming described, there's a great need to develop the next generation of monkeys with much larger numbers would allow us to do more high-throughput testing and testing of therapies. We have used in vitro fertilization of sperm from these monkeys to generate now quite a few of the F1 generation, and they're all showing this Shank3 phenotype. I also wanted to say that Guoping is developing efficient knock-in methods. Those are now working really at quite high efficiency. It's always been a problem making knock-ins compared to knockouts, but the knock-ins now seem to be working well. We now have animals being generated with a PV-Cre knock-in, which will give us cell-type specific targeting. This isn't just for the disease models, but also just for normal basic research in animals. It would be good to have many of the same genetic tools available in primates that are available in rodents, and I think those will be coming in line in the next couple years. So just in summary, we've successfully generated these animals with the Shank3 mutation. They do have behaviors that resemble some autistic-like behaviors in people. They have some circuit abnormalities which seem reminiscent of some that are found in autism, and we're now developing the next large cohort of animals that can be used. And again, this could only have come about as a result of our US-China collaboration, particularly with Guoping Feng at MIT, Yang Zhou, who's now at McGill, and our colleagues at SciAt, including Liping Wang, Zhonghua Lu, Huiwei Zhou, Shuo Yang at the South China Agricultural University, Andy Peng at Sun Yat-sen University, and all of their many colleagues. As we say, we had 25 people studying 5 monkeys. It was a big effort, but that's because we needed lots of types of expertise to get this project done. Thank you.
Thank you very much. Questions, please?
I have a question about your detour part. It's about the microcircuits. We have been working for a long time in the contingent negative variation associated to the Q. And then when the microcircuit is directed to the next target, when the microcircuit is directed in the same direction to the Q, then you have any change in baseline activity in this period, in the Q target period?
So you're asking when the microcircuit is in the same direction as the Q, is there a change in activity, let's say when there's no stimulus? Little or no change in activity unless there's an actual stimulus in the receptive field. I would say typically none. If you look at the LFP, sometimes you do see a microcircuit-triggered deflection in the LFP, even when there's no stimulus. But the bigger effects occur when there's a stimulus in the receptive field.
When the target arrives, or
what's that?
But when the target arrives.
Well, any stimulus, if there's a stimulus in the receptive field, any microcircuit is going to cause a change in activity. And we showed that in the control condition, where you actually do get a substantial visual response just by making a microcircuit in any direction. that causes a stimulus motion, which will give you a response. It's just that it's in the condition where the animal's directing a microsaccade towards that stimulus that you see all these big changes, depending on whether it was the cued attended stimulus and so on.
Can I follow that up for a second? Over here, Bob, on your left. So when you broke up the traces by the latency of the microsaccade, it seemed to me that the difference between the toward and away conditions was not that there was no response in the away condition, but that it was biphasic. There was an enhancement followed by a suppression. So when you add that up, averaged over all the saccade latencies, it disappeared. But there still was a modulation associated with each microsaccade. So what you seem to have was maybe a delayed suppression following microsaccades away, and I wondered if that was simply removing the target from the center of the receptive field in some way.
So, a couple things. So, yeah, just like we showed in the control condition, regardless of the direction of the microsaccade, you will get a change in activity just by making a microsaccade. The movements of the caused by the microsaccades, roughly about half a degree or so, were far smaller than the receptive field. So the stimuli aren't moving out of the receptive field or anything like that. And then what you basically, if you ask, so what's accounting for all the changes that you see, depending on whether it was in the cued direction versus another direction, you're going to get, it's a combination of a bunch of things. It's a combination of a visual response, it's a combination of some change in the LFP, and then it's some combination caused by presumably the corollary discharge to move your eyes in that particular direction. And so we're trying to actually sort all those differences out now, but the sum total of those effects is a big difference in the average firing rate. Bob?
Have you considered trying to turn off the microsaccade?
I think they want you to use the microphone, Bob.
Trying to speak
For all of us deaf people.
Yes, right, including me, right. So have you considered trying to turn off the microsaccades? I mean, they're clearly driven on exactly the same map as produces large saccades. It's accessible, it's retinotopic organoids, and it's called the spiricoliculus. and because at this point, it seems to me it's a correlation, but you could break it by turning off the microsaccades in a given direction, either permanently or transiently, and answer the question.
Yeah, I mean, we've thought about that. It wasn't so clear to me how easy it would be to suppress microsaccades by deactivating the colliculus. But if you think that would work, I definitely would want to add that to my list.
I think it would work. It's easy that I would stumble over.
Okay, interesting thought to try something like that. Yeah.
Again, microsaccades. When you said the classification or decoding gets better, so you had population responses taken from the cortex and then use a Bayesian decoder or something, and you triggered you took the phase when the microsaccade was targeted towards the target, after the target obviously had already been there. So which phase did you take in order to compare the decoders?
When the microsaccade is directed towards the attended stimulus versus away.
Okay. So you stratified those. And how long was the interval that you used for decoding?
I don't remember exactly. I think it was about 200 milliseconds, 250 milliseconds. Yeah. What it's beginning to look like now, again, we're in the middle of these analyses, something that we and others have typically seen, that the overall effects of attention are just an increase in the gain for the attended stimulus. But you look at a tuning curve, there's no change in the shape of the tuning curve. However, what it seems to be we're seeing now is that for the microsaccade towards the attended stimulus, it may very greatly enhance the preferred stimulus. But that on the return microsaccade, it's all of the unpreferred stimuli that increase their activity. And then when you add them together, it actually looks like the tuning isn't changing very much. But on the microsaccade towards, things actually, the fidelity of the stimulus representation may become much better. That's at least a in process and interpretation. Yeah.
So this work trying to promote monkeys with monogenetic alterations is fantastic. It will certainly help us to better understand major neurodevelopmental disorders affecting children, and certainly also the one or the other problem that affects adults, like genetically determined Parkinson's disease and so on and so forth. But most of the relevant psychiatric problems affecting adults, and the same holds for neurodegenerative diseases affecting adults, are not simple, don't have a simple monogenetic basis. So I'm wondering if we should not consider to identify monkeys with specific traits, screening large populations of wild-type animals. Any thoughts in this direction?
Yeah, so we've had discussions along those lines. One of the biggest problems is when you try to identify monkeys based on behaviors that you think have some relationship to humans. It's very difficult. I mean, you could always wave your hand and say, oh, this resembles a schizophrenic patient or something like that. You just don't know. But when you have a genetic lesion and then it leads to a particular behavior in a human and a particular behavior in a monkey, it's like, oh, now we know what behavior to look for. But without that, you're almost lost. I mean, even just phenotyping humans is really difficult, right? Saying which human is schizophrenic or not is not easy. So I'm not so convinced about trying to screen normal monkeys for relatively rare disorders. But I think some possible approaches are multiple genetic changes, which using like the cloning method that Mu Ming described, that might become possible to do a large number of genes. There are genetic lesions in most of the psychiatric disorders that are pretty highly penetrant. And the first thing would be to start with those and see what kind of traction you get in understanding the systems involved and so on. There's a possibility of xenografts of human material from human patients into monkey brains, and then seeing how those circuits develop. So I think there's some potential ways looking forward to the polygenic forms, but I agree, it's going to be a real challenge. The hope is that we will identify things in the monogenic forms that will then apply to the polygenic forms. But we don't know. It's just a hope.
Okay, if there's no further question, we thank again and go into the coffee break till 10:45, so good 15 minutes. Thank you.